Bryan Johnson says don't die. That's the goal, right? But what if one of the best ways to not die is to selectively kill some parts that refuse to die? A new study suggests that might actually be the case. That sounds extreme, right? Killing parts of yourself to stay alive longer. But here's the part most people don't think about. In healthy biology, cells are supposed to die. Programmed cell death, or apoptosis, is how the system is supposed to work. When a cell becomes damaged, dysfunctional, or potentially dangerous, it's supposed to remove itself. But some of them don't. These are called senescent cells. FOXO4-DRI is one of the earliest peptides that were designed to kill senescent cells. And lately, it's been in the news because it's been racking up some new results in aging blood vessels. As we age, some of our cells stop dividing, but they don't die. They hang around. They pump out inflammatory signals. They damage nearby tissue. They contribute to what we call inflammaging. These are called senescent cells. Some people call them zombie cells. A compound that selectively removes these senescent cells is called a senolytic. FOXO4-DRI is a lab-made peptide, a short chain of amino acids designed to do exactly that. Okay, think about this. Inside of cells that are senescent, there's a protein called p53. Think of p53 as a built-in self-destruct switch, and that's what triggers apoptosis. But there's another protein called FOXO4, and it binds to p53 and keeps that switch from being activated. It stops apoptosis from being triggered. Okay, so what the peptide FOXO4-DRI does is it blocks FOXO4 from binding to p53. It breaks that connection, and with that bond broken, p53 is now free to trigger programmed cell death, or apoptosis. The senescent cell dies, and it's removed by the immune system. Okay, so that's a little background for you. A new study came out last month that looks at this peptide in the context of vascular aging. Instead of focusing on whole body aging, the researchers zoomed in on blood vessels. Specifically, they looked at endothelial cells. These are the thin layer of cells that line the inside of your arteries. When these endothelial cells age, and particularly when they become senescent, your arteries stiffen, blood pressure rises, risk for heart disease and stroke goes up. The researchers tested FOXO4-DRI in 3 different settings. In wild-type mice that had aged naturally, in mice with accelerated aging that had been induced in the lab, and in human endothelial cells that had been pushed into senescence by exposing them to stress conditions. The researchers wanted to find the answer to several different questions. First, does FOXO4-DRI actually clear senescent endothelial cells? Does it activate the predicted apoptosis pathway? And does that translate into improved vascular function? Here's what they found. In aged mice, FOXO4-DRI reduced classic markers of cellular aging. It lowered inflammatory signaling and reduced oxidative stress. It disrupted FOXO4-p53 signaling and activated the apoptosis pathway in senescent cells. And perhaps most importantly, it improved arterial flexibility. In fact, the paper states that FOXO4-DRI effectively suppressed aortic aging and improved aortic function. In simple terms, older arteries started functioning more like younger arteries. However, keep in mind, this was done in mice and in cell cultures. There are no human trials or data yet. By the way, if you like this kind of breakdown, this is exactly what we do inside Longevity Lab. It's my free online community inside school.com, where we dig into new research and talk this stuff through in a lot more detail. And it's absolutely free. The link's in the description. All right, so why focus on blood vessels? Because vascular aging and arterial stiffening sit upstream of many of the most common age-related diseases. When your arteries stiffen, your blood pressure goes up, your heart has to work harder, the risk of stroke goes up, and kidney function goes down. Even cognitive decline becomes more likely. The endothelium, that layer of endothelial cells, plays a major role in all of this. When these cells become senescent, they don't just sit there quietly. They release inflammatory signals. They increase oxidative stress. And over time, they impair nitric oxide signaling and promote vascular stiffening. So the idea of selectively removing aging or senescent endothelial cells is not trivial. And if that mechanism works safely in humans, it could affect cardiovascular aging in a really meaningful way. And this is why this line of research is so damned interesting. Now, think about this. FOXO4-DRI is not new. It first showed up in animal research nearly a decade ago as one of the earliest senolytic peptides, and it generated a lot of excitement in longevity circles. However, since then, the peptide market has exploded. Today, you can go online and purchase just about any of the research peptides. Some of them are being studied seriously in labs. Some have early animal data, but most of the human data is anecdotal in nature. It comes from biohackers who are using peptides, not from human studies. FOXO4-DRI kind of sits in that gray zone. It has a very compelling mechanism. It has animal data, but there's really no meaningful human data. A recent article about this study on the Fight Aging website lays this out pretty clearly. And this article points out something important. Peptides that can be patented and controlled are far more likely to receive serious investment. That's because companies will fund large clinical trials only when they can own and exclusively profit from the compound. Peptides that can't be patented often remain in the realm of research and self-experimentation. That creates a really weird environment. The science, the business incentives, the regulatory system, and the consumer market do not always move together, as we all know. Researchers may understand a mechanism, but investors may or may not fund human trials because there's not enough profit in it for them. Regulators like the FDA require large, expensive studies. So research peptides can become commercially available to the public long before that human validation happens. This is why understanding peptides at a deeper level matters. It's one thing to read about a promising mechanism. It's another to figure out what to actually do with that information. And this is where a lot of people get stuck. They're watching videos about longevity, hearing about new compounds, seeing bold claims everywhere, but there's no real framework tying it all together. That's kind of why I created Longevity Lab on school.com. It's where we pull from all the different longevity theories and practices and try to frame a cohesive strategy, something that's personalized and that works. It's an active community of people focused on making smart, evidence-based decisions about aging. There's my free all-access foundational course called The Longevity Guide. That'll get you up and running with the basics of longevity. If you're interested in digging a little deeper, there's also paid content. There's a new premium tier where we take a really deep dive into topics like FOXO4-DRI. There's also a group coaching program called Primer on Peptides, and the next cohort starts on March 4th. But start with the free community. There's a link in the description. Join us there and continue the conversation. Okay, that's it for me. I'm out of here. Catch you guys next time.